Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I wanted to let you know that we are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called the Life Career Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life, and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey on drgpcr.com/career. We are also launching our GPCR consulting services. Soon you'll be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. The second edition of the Dr. GPCR Summit will be held between September 13th and 19th, 2021. We're planning a combination of live talks, pre-recorded talks, and live workshops. Visit drgpcr.com/summit for more information. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support us here at Dr. GPCR. All right, let's dive into our episode now. Hello, everyone. This is Yamina from Dr. GPCR. And today I have the pleasure of having with me Professor Ralf Jockers. And I hope I'm pronouncing your name properly. And that's what I forgot to ask you before we hit record, actually. He is a team lead of the functional pharmacology and pathology of membrane receptors at the Institut Cochin in Paris. Ralf, thank you so much for being here today.
Thank you, Yamina. Pleasure to talk to you again. So yes, indeed, my name was correctly spelled.
Thank you. I think that's kind of the reason why I kind of nailed it is because we've met previously and we have in common Nicolas Seveker, who was my PhD advisor. And I think he's the one who pronounced your name properly. So I learned that from him.
We are both German origin, so
yes.
Likely that helps.
That definitely does help. So can you tell us a little bit more about your current position and how you got into the position you're in?
So yes, I'm actually one of the lucky ones that have the pleasure to work full-time scientists in France. So there's this very nice system of scientists in CNS and INSERM. So since a couple of years, I'm one of them. So actually, 100% I can dedicate to my scientific research, and so now for quite a while I have my own group, and so I started after my postdoc. I came to France as a postdoc, and then everything went well. So I changed position. I didn't change the field, but so yes, and then I ended up with a permanent position and their own group and so on. So it was quite a dynamic and organic development. Mm-hmm. And now we are a group of 20 people, and it's quite a lot to manage every day.
Yeah, definitely. So you mentioned you're from Germany. Where in Germany, and how did you start, working in science initially in Germany?
Actually, I did my studies in Cologne. Mm-hmm. This was close to my hometown on the countryside. And then after my study, I went to Braunschweig, which is more in the middle of Germany, close, not so far away from Berlin, where I did my PhD in biotechnology. And then I decided not to go to the US. That was a little bit the time where doing a postdoc in Europe started to become an option. Mm-hmm. And so I decided to go. I was looking for labs all over in France, in Europe, and I ended up in France, Paris, which is of course a very attractive place to be. And so what helped a lot this was the time when the first EU fellowships, which then were termed Marie Curie fellowships were started to be granted. And so thanks to this, I ended up in France with this fellowship.
That's great. And when did you start working on GPCRs? Was it already as a PhD student in Germany, or was it when you moved on to France?
Yeah, that was actually part of the move. It was not only moving from Germany to France, but also moving field. Okay. So I didn't start at all with GPCRs. So I ended up with my postdoc on GPCRs, and then I stayed. But actually, I started biology of membrane proteins, so protein glycosylation, the ER. And then I actually worked during my PhD in biotechnological applications. So I stayed once again with membrane proteins, but here the idea was to apply this membrane proteins as biosensor for at that time herbicides.
Interesting.
And that was quite a strange project at that time. So I was the only one on this. But it was really cool because it actually it was inspired by the first by the Nobel Prize, which has been given for the first structural determination of a membrane protein complex, which was a photocenter of photobacteria. And so it was in collaboration with this lab at the Max Planck Institute of Biochemistry in Munich. And so the idea was that they knew how to purify this complex, and we used it on a biochip and to sense herbicides, because you have to know that some of the herbicides are actually inhibiting the electron transfer of this photosystem, and so that was my PhD. So yes, so I worked at that time on membrane receptors already, but not GPCRs. And then I wanted to do something more, let's say, clinically relevant. Mm-hmm. So moving to human receptors. And yes, and it's difficult to avoid GPCRs. So I ended up on GPCRs.
That's great. That's interesting. I didn't realize that herbicides, I never thought about the mechanism of action of a herbicide on a plant, and obviously it has to involve some kind of membrane proteins. Mm-hmm. And then what was your first time when you heard about GPCRs? When was the first time you thought, this could be a good project or a good career track that I should be going into?
Actually, since the beginning, I was always looking for something that was new and inspiring. So I was always fascinated by fields which were young, where you had the feeling that things can be done and it's not the end of the story. And in this sense, so yes, at that time, so I did my postdoc in the lab of Tony Strasberg, which was among those that cloned beta-adrenergic receptors. So he cloned it, in particularly the beta-3 adrenergic receptor. And so that was the time of, it was not the beginning, but was still the time of cloning the GPCRs. And then once you have the gene, that was, then you have a hand on a lot of things you can engineer. And that was a very pioneering time that I liked a lot. I had the feeling here you can do something, and there's something exciting to do.
And there is still a lot of exciting things to do in the field, for sure.
Yeah,
for sure. And now in your lab, what are you studying? You mentioned that you have 20 people. My guess is you're working on a wide variety of topics. Can you tell us a little bit more about the current work that you're doing?
Yes. So we have, let's say, a long-term interest in a specific group of receptors with GPCRs, which are the melatonin receptors. Let's say that's our workhorse. but work also in many aspects actually as an example for other GPCRs. So we have also other GPCRs running in the lab, but we also have we are not only GPCR people, we also have cytokine receptors. So we have let's say we are very GPCR centered, but not only. So for a long time, and still we are working on leptin receptor. Mm-hmm. Because we specialized after some time. We specialized in the membrane receptors involved in diabetes and obesity. So that's something that so we try to go into animal models. I know that currently there's a huge drive into molecular mechanisms, structures. Which G protein and so on. So very much molecular and cellular work. I think, and that's what we are doing. That yes, the bridge towards physiology shouldn't be forgotten, and that's what we try to do. That's why we need several people in the lab, because we are covering quite a large range of expertise.
That's very interesting that you mentioned, animal models. I think it's based on the different conversations I've been having with many of my guests. And yes, I think there are different approaches. Everyone has their favorite approach, but being able to encompass within one group various approaches that you can, at the end of the day, put together and ask questions about a receptor or about a pathway and look at it from different points of view, different angles is really important to better understand receptor function and hopefully draw that line between a receptor and a clinical effect. Mm. So since you've been working on various projects, various receptors, do you have a favorite GPCR or a family of receptors?
I mean, knowing the number of publications, so I cannot deny that melatonin receptors are my favorite ones. But yes, as I said, they are very, very good model for us, and we try to be original. So one of these attempts were actually when we recently proposed or get a hand on an alternative, most likely signaling mode. Actually, one of these receptors, which is still an orphan, is GPR50. So we don't know the whether and through which G protein or beta-arrestin molecule it signals, and so confronted to this problem, so to say, we were also exploring other options, and among these options, actually, we made an interesting observation that the C-tail, which is quite long for this receptor, is proteolytically cleaved.
Interesting.
And so at the end, we could show that upon cleavage, like other family receptors, actually this becomes a transcription factor or cofactor. So this is the cleavage of the C-tail, then translocates this C-tail into the nucleus, and then it activates gene transcription. So you see that it's a completely unusual signal transduction mode for GPCR. Of course, we are still looking for the ligand that could trigger this, but that sounds quite interesting for me. So that was quite. This was one of my favorite projects, and it's still one of my favorite projects.
That's interesting. It's a very different way, as you mentioned, of a receptor to exert its function. How did you make that observation? What were the experiments, or how did you even start working on GPR50?
GPR50, so it has the highest sequence homology with the other receptors that actually bind melatonin, MT1, MT2. And so it was a natural member of the same cluster. And yes, the observation was very trivial. It was, as usual, it's tools who are driving us. So in this case, it was we developed antibodies against this receptor. which was is one of the examples where the success was quite good. Antibodies and GPCRs are things that they don't like each other usually, but for this one, since it has a very large C-tail, it was not a major problem. And then we discovered in tissues and so on that yes, there's proteolytic cleavage of this C-tail, and then we try to understand how is this an artifact or is it real? And it turned out to be real, and it turned out to be functionally meaningful.
That's so interesting. And what kind of genes was it targeting? Was it inhibiting, or was it increasing the expression of specific genes?
Actually, we did in parallel a proteomic screen for interaction partners of this C-tail. And we ended up with transcription factor TF2I, and then we could show that this is early CFOS gene transcription, and actually then we did some what was it? We did some gene expression studies, and we could show in cells which expressed or not this C-tail that there were something like 2,000 genes which were actually differentially expressed. We did gene array. This is the way that the word that I was missing, to gene array, gene expression array, and so it's huge. It's huge. Of course, this is a system where you overexpress the C-tail, so you are pushing the limit. That's for sure. But yes, the impact is massive, and so now we are trying to find out in animal models how this what happens in vivo with these transgenic animals.
Of course, and it's such an interesting. It's interesting because I was talking to Professor Randy Hall, I think last week when we recorded our podcast episode, and he was mentioning that when he joined Bob Lefkowitz's lab, he decided to ask the question about what use is there for a GPCR to have a C-terminal tail. Mm-hmm. And it turned out that, yes, it was very important, and we all know that, but this is a unique property of this GPR50 C-terminal tail. What tissues is this receptor expressed in? Is it the same tissues as MT1 and MT2 in the skin, or how does that?
Seems to be quite different. So it's quite broadly expressed in periphery, but the let's say the prime site of expression are in the brain, tiny sites, which are actually a unicellular layer in the third ventricle. Actually, it's a border between the brain, the hypothalamus, and the liquid in the third ventricle. So it's a very discrete site where it's highly expressed, and it's actually a marker gene for these cells, the tiny sites.
That's so interesting. That may mean that it's their control of this receptor is very important, and their role is very specific to this brain area. What type of mouse model are you looking at when studying this receptor? Is it a disease model or just a?
So for the moment, we have knockouts, and actually we also generated human knock-in mice. And so yes, the models that we apply for the moment are rather towards metabolic phenotypes. Indeed, knowing that the hypothalamus is also very important for energy and glucose metabolism. That's exactly.
That's such an interesting receptor. It never amazes me to learn about GPCRs that do very funky things that turn out to be, nicely controlled in the body, and Mother Nature knows pretty much best what she has to do. Mm-hmm. What other disease areas is your lab interested in? You mentioned metabolic diseases, you mentioned diabetes, type 2 diabetes. Any other topics that you're interested in exploring, or are you currently exploring?
We worked for a while. Also, melatonin receptors. They are involved. There are drugs on the market on sleep and depression, but that's actually not our field of expertise. Mm-hmm. We we are, I have to say, we dived slowly over the last couple of years a little bit into something on the border between metabolism and Alzheimer's disease. So we are getting more and more interested in the metabolic component of this of the Alzheimer's disease. So there seems to be a quite important overlap and cross interactivity. So we don't know who comes first, huh? But
Yeah.
clearly there seems to be a significant overlap, and we are trying to find out whether we find new targets. So actually, general idea is maybe metabolically important receptors could be interesting targets, new targets for Alzheimer's disease.
Any receptors you care to share with us?
I can say that, for example, when you treat people with insulin, or when you treat people with GLP-1 receptor agonists,
Yeah.
they have clear beneficial effects. So that this kind of receptors that are most likely at some point important.
I think controlling insulin levels in the body is one of the most important things. It's interesting because there's a podcast I like to listen to, and it's from this professor at Stanford, Andrew Huberman, and it talks about, how to translate scientific findings into everyday life. And there was a podcast about insulin and insulin control and cortisol. And I feel like that's very important to control that glucose level and that insulin level in the body in order to promote health. And there's still much to learn about the effects of high glucose, other than having the long-term effects of those.
Absolutely.
That's so interesting. So you mentioned tools, and you mentioned better tools to understand receptor function, for example. What kind of tools do you use in the lab? What kind of type of experiments? You have animal models, and what I'm getting to is, and I'll put it out there, is the Brett tools that you actually even shared with me back in the day. Are you still using that type of cellular basic functional tools?
Yes, absolutely. So yes, it's never enough to insist that you can have the smartest ideas, but if you cannot, if you don't have the tools to show what you have in mind, that you have a problem. So yes, these tools are very important, and that's why in the early days, indeed, beginning of 2000, we developed this, and we worked a lot together with Michel Bouvier. And so since then we are using them, we are developing them, we are having variations. So yes, we still use them. We diversified. We are also using a lot TR FRET, time result FRET, which is also very, very nice technique. The complementation assays, the non-loop complementation assays. So all this is very, very, very nice. So initially we used this to measure protein-protein interactions. That's, I mean, dimerization was, of course, still an issue. But now, yes, all these tools developed also very nicely into ligand receptor interactions. So that there's a lot of diversification that they're really useful, these tools. And in the meantime, yes, we developed also some proteomic tools for GPCR. So that was also very interesting. So we do less at the moment. We are trying, actually, when we have a program for deorphanization of GPR50. So then we are looking at new techniques that allow us to measure ligand binding without labeling. So label-free techniques. So I think this is also very, very interesting new field where you don't have to modify your protein, and because then you're much faster. You can work with very small molecules that you don't have the risk of modifying the function. You can work with biological fluid samples. So that's something I think that it is quite interesting that we are trying to push a little bit at the moment.
Well, label-free is definitely an advantageous technology, and I think it can be very useful. And I love the idea of using it with biological samples. Then you can go really fast and go and read as many plates or as many compounds or as many samples as you'd like. Let's get back to GPR50. So you mentioned, well, it's an orphan GPCR, and there is a sequence homology with the melatonin receptors. Does it bind melatonin?
So far, nobody saw melatonin binding to this receptor.
That's interesting. And so there is absolutely no clue of what type of ligand it might be binding.
Actually, when you the structure of the MT1 and MT2 melatonin receptors have been solved. So when you do just by homology modeling, you model the structure. It looks very similar. Even the binding pocket is very narrow, very small. Actually, you have to know that for the melatonin receptors, that there's something very particular. They have two entry pockets, one from the standard one from the top, but also a lateral one that is not seen so often. So the lateral one that means from the membrane environment, seen also for the CB1 and a couple of other lipid receptors, but that's quite unusual for a molecule that actually comes from the extracellular side, so which is not a lipid. So that's also something that is quite striking. It seems to exist also for GPR50. So that's something that we are looking at the moment. So to find this ligand for this receptor.
Interesting. And so we've talked a little bit about tools. What do you think? What kind of tools are we still missing in order to speed up the orphan GPCRs, or even just quote on air quotes, advancing the field and better understanding the function of GPCRs of interest?
That's a difficult question. I think looking back, I think there was a huge hype and huge success in deorphanizing the receptors, but since the last 10 years, actually, that's stopped quite a lot. So there is something. There are not so many successes, and since then, I think the successes are rather one by one. So it's more case by case. There are some success stories where with peptide receptors, the deorphanization, or the paper from David Gloriam, where they matched the possible peptides from the human genome with still orphan receptors. So they were able to have this deorphanization of several of them. But apart from this peptide story, actually, it looks like a one by one struggle. Always with the possibility that maybe not all of them have, let's say, kept the capacity to bind to a ligand. So there might be a subfraction of them that evolved towards other functions. And that actually we tried to show with GPR50 and others, where actually so GPR50 is a good case actually, where we showed that it's by dimerizing or by interacting with other membrane receptors is regulating actually the function of the other one. It's more like if you don't know what is the function of myself, you might know the function of my partner. And so that we could show for the MT1 melatonin receptor. We could show for the actually TGF-beta receptor. So we picked this up in a proteomic screen and could then show that yes, it's really happening in real life and is actually modifying is important for cancer. So TGF-beta is an important player in cancer, and actually GP50 is modulating, is actually constitutively activating the TGF-beta receptor. So yes, maybe some of these orphan GPCRs, they have just evolved towards another raison d'être. Yeah. They certainly, as we actually for GP50, we have evolutionary evidence that it at some point bond to melatonin, to the ligand, and then lost this capacity. But of course, then you have to find another reason to be kept alive during the evolutionary pressure. And so maybe these other functions are one of them.
You touched on an important point is keeping an open mind and knowing that, yes, we're looking at GPCRs because these orphan receptors look like GPCRs, but keeping an open mind and thinking, oh, they may be functioning in a different manner. They may be modulating the function of other proteins, or even are there to, carry, for example, that C-terminus tail of GPR50, and the activation of another signaling pathway might lead to cleavage of that tail, but it has to be somehow expressed in the cell, which is I find a very interesting concept, and I think we should be digging more deeper into what does it do, and it might also. Please go ahead. I was going to say
actually for some time, of course, when you start to sell a concept, you have to push to the limits. So for some time, I was rather advertising this as one or the other. So there are those who have evolved towards other functions, and the others are the more traditional GPCRs. But I'm more and more thinking that actually there's no reason that they're mutually exclusive. And there are actually examples for interesting examples for this. There is, for example, the ghrelin receptor that has several, let's say, several modes of actions. So it's a receptor which has a high constitutive activity. So that's already one thing. So you don't need a ligand for this. The other thing, there is a ligand. So ghrelin it has a ligand. You can regulate it with a ligand. So far so good. But in addition, when it can heterodimerize with dopamine receptors, I don't remember whether it was D1 or D2. I think it was D2, where actually it's regulating the D2 receptor in this complex, and this is independent of ghrelin. So here you have another example, and that this receptor can satisfy all the different modes, and without being an orphan. But yes, depending on the context, it might function in one or the other mode.
Yeah, which is interesting when you think about it, because then you realize that, again, going back to Mother Nature, she knows best, and why come up with a new gene or why come up with a new protein when that one protein, depending on where it is localized, where it is expressed, and who it is expressed with, you can modulate the function that way. That's really fascinating. I think it's a really nice way of looking at it. You mentioned, the interest into tying in your functional work or the work that you do in the lab to clinical outcomes. What do you think is missing, for example, for your team to say, well, we've come up with a molecule, we've come up with something that we can push towards a more clinical application?
Maybe I can introduce you another aspect that I found interesting that we are working on, actually since a while, where I think my group or the GPCR field can make a significant impact, which is the field of GPCR variants.
You mean splice variants?
No, not splice variants, but just genetic variants. So it can be splice at splice site. can be in the coding region, can be in the regulatory region. So it's genetic variance. That's something that we started in 2012 with the melatonin receptor, which actually that was let's say a unexpected finding that the geneticists they're unbiased, so they look for risk genes for a specific disease or a specific trait. And at that time, there were 3 back-to-back paper in Nature Genetics, where they did the first GWAS studies, gene-wide association studies on for diabetes and associated traits. And so one of the genes that really come up, which had a very high score, was a variant in this gene coding for the MT2 receptor. And that everybody was, well, what's that? We never heard about this. And then that sounded interesting for us. And so this was like usual was a variant, a SNP, which was not in the coding region, but it was in the intron, so nobody knew really what it does. And so our idea was to have a closer look in other variants in the coding region, and that's where we started this enterprise. So looking with the geneticists for this variants in the human genome, then see whether they're associated with this risk, and then try to understand why. So that's the big panel of breadth sensors and other tools to characterize where's the problem. So then you see, okay, my variants, my mutants have rather loss of function, gain of function. Yeah. That's the first thing that you're interested in. And then if you have enough of these variants and if you have enough work capacity, then you might ask the question, what is the precise mechanism? So is it a specific pathway? Because then with the idea that at the end all this information can give you a precise idea what you are actually which kind of drug you are looking for. Are you looking for something that activates or inactivates my receptor generally, or one pathway or the other? So that then you have a quite direct link to something that to tailor drug design.
It's so interesting. Did the mutation in the intron of MT2 alter the receptor expression or function, or what did it result to? You said it was a SNP.
Yes. We didn't work on this directly, but those who tried to work on this, they saw a change in the mRNA level expression. So yes, but actually it's still controversial. It's not really clear whether, because we don't know whether actually where is the expression of this gene is most relevant. Is it in the periphery, like in the pancreas?
Yeah.
Or is it in the central nervous system, where actually melatonin is regulating the circadian clock? And this regulation of the circadian clock also predisposes to metabolic diseases. So there's still a debate on whether this is, and also the change was twofold. But yes, it's difficult to judge whether it's enough. But this is the current hypothesis for this SNP in the intron. Yeah.
I like the idea to think broadly and think about, yes, the SNPs or any genetic variations of receptor genes as a way to kind of better understand what they would do in specific diseases, and at least even pinpointing out if any genetic variations predispose people or patients to some diseases, which would be really interesting to know, because then you could directly target these specifically mutated, or not mutated, but genetically altered genes in that specific population with compounds that modulate that receptor function that is affected.
Yeah, exactly. And of course, this is very personalized, but it's something that we are getting to. I mean,
Yes.
it's not you are not targeting the big diabetes market for everybody. So
Yeah.
that's something a little bit different. But it actually in this study, it was also interesting. It was not only all about finding the right drug, but it helped us also to give since melatonin, so it's involved in circadian rhythm, and then your next step is that it's important whether you have a disturbed circadian rhythm, whether you have a disturbed sleep, whether your eating behavior is in matches with your circadian rhythm. So there's quite a range of behavioral components that you can then study with these patients, and then if there's a misalignment, you can give them quite precise advice to change what should be changed in their daily routine. And that's also quite immediate way to translate the findings.
Yes, and that's interesting. And it doesn't involve a pill. It just involves adjusting sleep, adjusting eating habits in order to give the body that that rhythm that it can sustain in the long term, which I find really interesting as a topic.
At least to help to overcome this weakness that you have because of your genetics.
Yes, definitely. As I was listening, I'm very interested in better understanding how the body functions, the circadian rhythm. Having a newborn at home definitely affects the sleep patterns. But I've been listening again to these podcasts when they're talking about exposure to light first thing in the morning to get you. Get your brain basically and your circadian rhythm understand, okay, now it's this amount of light, it's morning, and doing the same thing in the evening when the sun goes down to, again, alter your rhythm and associate it with, some sun coming up and sun going down and unwinding in the evening. And I think even the eating habits also play a role in that. Even when you drink your coffee in the morning plays a role in your wakefulness. So apparently delaying your first coffee by 2 hours after you wake up allows you to be more alert longer during the day.
Okay.
Which is very difficult to do, but there is scientific evidence that actually the adenosine receptors, the adenosine binds better the adenosine receptors on a regular like a normal physiological pattern. But if you drink coffee, you compete off the adenosine with the caffeine, and it allows you to stay more alert. I did not try it yet. I find it very difficult not to start my day with a coffee. But there's one way of altering your physiology without taking any drugs in that sense. Although coffee can also be a drug at that point.
It has also some beneficial effects, huh?
It does beneficial, and also a lot of social effects, or at least before COVID hit. I think that also changed everyone's life. So you mentioned that you have about 20 people in your group. What is the distribution of people? You have a lot of trainees. The reason I'm asking this is because the next question I want to ask you is what would be your advice to young scientists who want to contribute to the field?
Actually, it's a mixture of students, postdocs, and so in France, with this system of permanent scientists, CNS and INSERM. So we are also lucky to have we have 5 permanent scientists. So we have a mixture of all of these people, which is quite good, and technician. Also, so it's I think the mixture is good.
It's a nice balance then.
Yeah.
So as young scientists prepare to enter the field of GPCRs, or even just working in the lab and becoming scientists, any advice for them?
Yes. Maybe they will listen, or maybe they shouldn't listen to everything. So yes, I would say that many, especially in France, where people are still young and a little bit unexperienced when they are coming to the lab. So the young people that they try to naturally, they try to go for the obvious, to go slowly, to go to the traditional tracks.
Mm-hmm.
Well, I think yes, my advice would be don't be afraid. Don't be afraid to ask trivial questions, to ask questions, to think about the impossible. So I think yes, scientist has to be first of all curious and ask questions that maybe nobody asked before, and also ask big questions. Sometimes people are, especially students who don't know, of course they are young, they don't dare to ask big questions. But I think that many people maybe ask only incremental questions. So I would encourage the young scientists to ask questions, relevant questions, and don't hesitate to engage into new territories.
I think you mentioned, the hesitancy of younger scientists to ask bigger questions. I think that comes with experience, and experience brings confidence. But just because you haven't been at the bench or haven't worked for 30 years in the field, that doesn't mean that your questions aren't valid. And I think the whole point is to ask those questions so that you can discuss them with fellow scientists and go with your gut.
Yeah,
Definitely.
it can be even the opposite. Huh? I mean, very often you have to be naive. Maybe, and this naiveness makes you to ask or to go for something that you wouldn't go if you would have more experience. So
Yep. Definitely. I think it has its advantages to ask questions that you think are interesting, not necessarily relying on your experience, but just being curious. Curiosity, it's a good thing, I think.
and at some point, of course, the role of the supervisor is then to put all this together and not to lose people, to get them lost. So to give guidance. But yes, I think at the beginning there's good reasons to be a little bit crazy.
So one last question for you, Ralf. We've talked about the different projects that you've been working on and the different interests in the lab. Were there any moments during your career where you stumbled upon a result or discovered something that kind of changed your way of thinking about the field, which is something I call the aha moments that changed your life as a scientist?
I think it was quite linear, but maybe the biggest changes were at the very beginning. Actually, maybe the decisive moment were when I asked me the question, "What do I want to do? Do I want to become a scientist?" With all the naivety, not knowing what it really is. But yes, then having people that really are inspiring you, or going for something else. Huh? Actually, I was very early on hesitating between three options. So either becoming musician.
Interesting.
Musician. I invested quite a lot of time in playing trombone. Or becoming an engineer like my father.
Mm-hmm.
And I was already actually registered to university until I met a scientist, a friend of me, who convinced me that biology or become a researcher might be the thing that I like most. And so yes, I choose between these three very different destinations. So yes, maybe the aha was to speak to people that are really inspiring, and that are inspiring enough to drag you into something, into this big decisions of your where you want to go into in your life. So, and that was quite and yes, I don't regret.
That's great. That's great. Do you still play the trombone?
Less and less, I have to say. And the bad thing with trombone, if you don't exercise, it's not like piano where you always just have to sit and at least at some level play. Trombone, yes, you need to train your lips, otherwise you are tired a lot. So you need to have time and to have training every day. So let's see, maybe later.
Yeah. I think it could be also a nice way to let your brain focus on something else while you're playing the trombone, and think about your brain kind of take it in an automated fashion and think about projects. I mentioned this a lot of times during podcasts, but the best ideas I had were not while I was working in the lab, was walking the dog or doing something completely different. And that's when my brain had the time and the break to come and find the answer I was looking for.
Absolutely, yes. You have to have something else.
Yes.
To avoid your brain, knowing that your brain is always working in the background, huh?
Yes, which is scary, but at the same time really fascinating, knowing that even when you're asleep, your brain is actually still working 24/7. All right, thank you so much, Ralf, for your time. I really appreciate it. I hope you had a good time. I did.
It was a pleasure.
And with this, I'll talk to you soon. Thanks.
Bye-bye.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Duran, and Ines Pinero. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us a testimonial at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.